Hello students, welcome to part 1 of our 2019 Biology State Questions. Let's begin. A student performs an experiment to study the transport of water in and out of cells. He uses clear, semi-permeable plastic bags to represent the cell membrane. The student places 3 plastic bags of the same size, each containing a 1% concentration sucrose solution, into 3 different beakers. One beaker contains distilled water, another beaker contains a 3% concentration sucrose solution, and the third beaker contains a 1% concentration sucrose solution. The diagram shows the student's experimental setup, the plastic bag before it is placed in the beaker, and the appearance of the bags an hour in each solution. So let's look at our picture. Experimental setup and results. So we have a 1% sucrose solution. That means we have 1% sugar and we have 99% water. Okay? So we go over here, we put that bag with 99% water into distilled water, which is 100% water, right? And we know with osmosis, water moves in and out of the cell. Water goes from a high concentration to low. In this case, since more water is on the outside than the inside, then the water will go inside the bag. Over here, we take our 99% water and put it in a solution which has 97% water. Now, more of the water is inside the cell, okay? So it wants to go out of the cell to go where there's less water. And if we look at the third beaker, we have 99% water being put in a solution of 99% water. So it remains the same. Which statement provides a conclusion about the transport of water in and out of cells based on the results of the student's experiment? A. Water moves freely both inside and outside the cell, independent of the solute concentration. This is not true. Water does not move in and out of the cell by itself. It moves in and out of the cell based on the concentration of the solute in and outside the cell. Let's look at C. Water transports solutes out of the cell to balance the percent concentration of solutes both inside and outside the cell. The water is not carrying the solutes, it's the water that's moving in and out of the cell. D. Water moves into the cell and solutes move out of the cell to ensure their concentration is balanced both inside and outside the cell. Again, it's not the solutes that's being transported, it's the water. So the correct answer is B. Water movement into the cell is dependent on the percent concentration of solutes both inside and outside the cell. And that's what we saw in our diagram. Let's continue with question number 2. An isolated population of seals experiences a bottleneck event over a period of time. Even after the bottleneck event ends, the genetic diversity of the population still shows its effect. A. Use the Connect Line button to add a line segment to the graph showing the change in genetic diversity during the bottleneck event. B. Use the Connect Line button to add a line segment to the graph showing the change in genetic diversity after the bottleneck event ends. Remember, when we talk about a bottleneck event, we're talking about a change in the allele frequency of a population, not by natural selection, but by a chance. And I always tell you to remember the man that stepped on the 10 bugs, right, killing 3 of them by chance and changing the allele frequency of that population. So we know when a bottleneck event happens, the genetic diversity will reduce drastically. And then after the bottleneck event ends, that change in genetic diversity will continue, but slower. So let's indicate that with this chart. So it's at a point, connect line. Okay. So there we're showing that the bottleneck effect really drastically changed the genetic diversity. Now what happens after the bottleneck effect? The change is we're still getting less genetic diversity, but it's not as a steep slope, as a slope. Okay? That's all there is to this one. Okay, let's look at question number 3. The table shows the derived traits of 5 plant groups. And we have the 5 plant groups over here. And we have the derived traits. We have flowers, seeds, stomata, and vasculature. Now, what's our question? Which trait was the earliest to appear among the plant groups? Okay, this is an easy question, right? All we have to do is interpret this table, okay? The trait that exists in the most plant groups is the trait that was the earliest to appear. So if we look here, we see the flower just appeared in 1 plant group, seeds appeared in 2, the vasculature appeared in 3, but the stomata appeared in 4. Therefore, the correct answer is the stomata. And a stomata is a little opening in a leaf that allows the exchange of gases. See? Not a hard question at all. Question 4. A table of organisms and traits is showing. Use the data in the table to complete the cladogram. And our organism traits are amniotic egg, backbone, eggs with shells, 4 limbs, and hair. A. Move the correct organism letter into the appropriate blank box at the top of the cladogram. And B. Move the correct trait label into the appropriate blank box at the bottom of the cladogram. Okay, here's our cladogram. Here we have C, we have backbone, we have blank, amniotic egg, we have blank, and we have hair. So what we need to do is find who just has the backbone, and that is F. F does not have any more traits, just the backbone. What's going to be our next one? Okay, so we see here that organism B has the backbone, so he gets over that hurdle, and he has 4 limbs, but he doesn't have amniotic eggs. So now that tells us we can put amniotic 4 limbs here before the amniotic egg, and we can put B over here, right? Because B has a backbone, he can jump that, and he can have the 4 limbs, he can jump that. Now we need somebody to jump the backbone, the 4 limbs, and the amniotic egg. So let's see what we have here. We have A can do the backbone, A can do the 4 limbs, A can do the amniotic egg, and A can do hair. So we can put A up here, right? Now, we can look at another one here. We got E. Okay, let's look at E. He has a backbone. That's good. He has 4 limbs. Excellent. He has the amniotic egg. That's good. And he has hair. So we can put E here also. So what we're left with is, as a trait, eggs with shells. And let's see who meets that category. It's D, right? D has a backbone. D has 4 limbs, right? So he can jump that. D has the amniotic egg. D does not have hair, so he's not going to go up that path. He does have eggs with shells, so D is here. And that's our cladogram. And remember, these two are closely related because they have the closest common ancestor. Let's look at question 5, part A. A geneticist identifies cell types in 4 sexually reproducing organisms that display mutations. The results are shown in table A. Plus symbol means a mutation was detected. A minus symbol means no mutation was detected. So if we look here, we see organisms tested, LMNO. And we have skin, stomach, and gamete, okay? The question is, which organism will pass the mutation to their offspring? When we look at our chart, we see skin, stomach, and gamete. Okay, we see a mutation in skin for M, mutation in stomach for N and O, and mutation in gametes for L and M. Okay, so the question asks which organisms will pass the mutation to their offspring. That's easy. Skin and stomach are known as somatic or body cells. Mutations can only be passed on in the gametes. And remember, gametes are sperm and egg. So the answer to this, which organism will pass the mutation to their offspring, is L and M. Now, when we look at part B, which statement describes evidence to support your choice in part A? Well, the very first one. Offspring genotypes are determined by gametes. They're not determined by somatic cells. When there's a mutation in the gametes, okay, that is passed on to the offspring. Question number 6. Chemosynthesis is a cellular process used by organisms in deep sea ecosystems in the absence of light. A chemical equation for chemosynthesis is shown. Here's our chemical equation. Okay? Now, remember, chemosynthesis is different from photosynthesis because photosynthesis uses light to make glucose and oxygen. Chemosynthesis doesn't have the luxury of having light. It's deep in the bottom of the ocean, and it uses chemicals. So how is chemosynthesis different from photosynthesis? Chemosynthesis requires carbon dioxide. Well, we know photosynthesis requires carbon dioxide, right? It includes water, carbon dioxide, and sunlight to make the glucose and the oxygen. B, chemosynthesis is used by autotrophic organisms. Duh, so is photosynthesis. Chemosynthesis produces energy in the form of sugars. Well, there's your sugar right there. Photosynthesis does that too. D is the correct answer. Chemosynthesis harvests energy from other chemical compounds, whereas photosynthesis harvests energy from the sun. Question number 8, and this will be the last question for part 1 of the 2019 Biology State questions. So let's get it going. The European corn borer is a pest whose larvae causes damage to corn crops in the United States. Pesticides can be used to control the larvae. As an alternative, scientists have developed a genetically engineered form of corn called the Bt strain. It produces a toxin in the cell called Bt, which is poisonous to the corn borer larva. Farmers who plant fields of Bt corn are required by law to plant fields of non-Bt corn nearby. Here's our question. Which statement explains why farmers would be required to plant non-Bt corn near the Bt corn? A, to provide controls for Bt toxin experiments with corn borers. This is incorrect. While studies may be done using some farm fields, this would not explain why all farmers would be required by law to plant both types of corn. We're not doing an experiment here. B, to keep corn borers from cross-pollinating Bt and non-Bt corn. This is incorrect. Planting fields of Bt and non-Bt corn near each other would not prevent corn borers from cross-pollinating the two types of corn. Let's look at D. To prevent movement of corn borers from Bt to non-Bt corn fields. Planting fields of Bt and non-Bt corn near each other would not prevent the movement of corn borers between the two fields. It would enable the movement by placing the two fields in close proximity. So our answer is C. It's a slow reproductive selection for a Bt toxin-resistant corn borer. Why? The presence of the Bt toxin would encourage natural selection for individuals that are resistant. In other words, we could have insects that would become resistant to the poisoning. Planting a field of non-Bt corn would help slow the process by ensuring that heterozygous individuals would remain in the population. Friends, this is our last question, and I hope you enjoyed it. Thank you.